March 25 exam was an easy exam. The first question was about movement of molecules in and out of the cells. Okay, this is a cell. This is the cell membrane. Okay, here I have high concentration of molecules in P. Here I have high concentration of molecules, and here is low concentration. So the molecules are moving from high to low. But which process is this? This is diffusion. Here I have low concentration of molecules, high concentration of molecules. It is moving inside down the concentration gradient. So what is this? Again is the concentration gradient. What is this? This is active transport. Okay. Arrow P represents diffusion of oxygen molecules. What is diffusion? It is the net movement of molecules down the concentration gradient or from high to low. And don't forget this part, it's as a result of random movement of particles. Well, why does diffusion happen? Diffusion happens because the molecules in liquids and gases, they are already moving. So what type of energy is needed? It does not need external energy, but why does it happen? Because of the movement, which is the kinetic energy. Kinetic energy. The molecules move, so they diffuse. Carbon dioxide molecules also move by diffusion. What is the name of the process that produces carbon dioxide? The cells produces carbon dioxide. How does they produce carbon dioxide? Aerobic respiration. Draw an arrow to show the direction of diffusion. Carbon dioxide, but how does it diffuse? From inside the cell to the outside. That direction of diffusion of carbon dioxide. Q represents another type of movement. Very easy. Low concentration to high concentration using energy. So this is active transport. I chose active transport. Why did I choose it? What is the explanation? The molecules move against the gradient using energy from respiration. And this was in the diagram. Okay. In the figure 1.1, R moves from the lumen of the intestine, of the small intestine, into the epithelial cell. R from the lumen of the small intestine into the epithelial cell. Why R cannot be starch? Why cannot it be starch? I have various reasons. Number one, starch has been already broken down in the small intestine. It does not stay as starch. Amylase enzyme breaks down starch into maltose sugar. Number two, starch is too large. It cannot pass through the membranes and it is insoluble. So why R cannot be starch? Because it is insoluble, it is too large to pass through, it has been already broken down. I need two from these. Type. This is another question on the movement in and out of the cells. And this question we had it before in one of the papers. Red onion cells. Red onion cells. This is a plant cell. So the white lines are probably the cell walls and the rest of the content is inside. This is the same red onion cell after being immersed in salt solution. What happened? It shrunk from the inside and the cell membrane separated from the cell wall. There are gaps between the cell membrane and the cell wall. Okay, I understand. He put it in salt solution. Salt solution, يعني low water potential. Concentrated salt solution. And inside the cell was high water potential. So the cell lost water, it became flaccid and plasmolized, cell membrane torn off away from the cell wall. All of this I could say, according to the question. So let's see the questions. Using figure 1.2, 1.3, describe and explain. Describe and explain the difference before and after immersion. Before immersion, description. Before immersion, it was a turgid cell. The vacuole was expanded. fully covering all the cells. It was a turgid cells, the cell membrane pushing on the cell wall, and the turgor pressure, it has turgor pressure. But what about after? I will describe first. Cell membrane torn off away from the cell wall, creating gaps between them. The vacuole shrunk, okay? Uh, this is the description. It's a plasmolyzed cell, no pressure anymore. All of this is the description. يبقى before immersion, the cells are turgid, membrane pushed against the cell wall, creating turgor pressure, while after immersion, they are flaccid. Cell membrane pulls away from the cell wall, creating gaps, so it is plasmolyzed. طب what is the explanation بقى? Why did this happen? Why did this happen when you placed it in salt solution? Because the salt solution has a lower water potential. So the water is lost by osmosis down the potential gradient from high water potential inside the cytoplasm to low water potential in the salt solution through a partially permeable membrane. Of course, this all occurred by osmosis. By osmosis. Question 2. Internal body temperature. Using the information, calculate the temperature range. range بتاع الجسم من كام لكام؟ ما بين 36 و 38. calculate يعني في حاجة يعني I need to calculate something. The range in the body temperature is between 36 and 38. So I need to calculate something. What is the range? 2 degrees Celsius. 38 minus 36, it is 2 degrees Celsius. Draw a line to show the set point for the internal body temperature. What is the set body temperature? What is the normal body temperature, the average? It is 37. I will draw a horizontal line on 37. This is the set body temperature. Maintenance of internal body temperature is an example of homeostatic control. What is the mechanism for homeostasis? How does homeostasis work? If the temperature increases, it returns it back. If it decreases, it returns it back. What is this called? It is called negative feedback. This is the mechanism by which homeostasis occur. Negative feedback. Okay. On the figure, region F shows a change in the body temperature. This is a tricky question. Explain how the body causes the change in the body temperature. Let's see region F. What was happening in region F? The body temperature was low, so the body did some mechanisms to increase it. Body temperature was already low, and then the body did some mechanisms to increase it. How does the body increase its temperature? I'm not talking about the external temperature. يعني if the external temperature is high, what will happen? لا. I'm talking that the internal body temperature is increasing. It was low. I was cold. I was feeling cold, and I'm increasing my temperature. What will the body do to increase the temperature بقى? Vasoconstriction, skin arterioles constrict, the shunt vessel dilates to keep the blood away from the skin, prevent loss of heat by radiation. Also, shivering occurs to release heat energy from the contraction of muscles. الhair erector muscle contracts. الhair is upright to trap a layer of insulating air. Figure 2.2 shows a cross section of the human skin. The labels of the skin. Which letter produces the sweat? This structure, okay? This is the structure that produces the sweat, that is opened on the outside. This structure is called the sweat gland, and it is opened on the sweat pore. يبقى which produces sweat, it is the sweat gland. طب what are C? The muscle holding the hair, it is the hair erector muscle. D is the sensory neuron, the neuron that is going downwards, it is the sensory neuron. Okay. Question three. It shows the percentage of water in different structures of the human body. Percentage of water. The mean mass of the human eye, is 28 grams. Using information, calculate the mass of water in the human eye. طيب. The mass of the eye is 28 grams. The water is 95%, so I'm just gonna calculate it on the calculator. 95% times 28 grams, it will give me 27. If there is decimal points, you need the number to be two significant figures only. 27. What is the importance of water in the human body? In the human body, it is a solvent for metabolic reactions. It is a solvent for digestion. It helps in digestion, dissolves the nutrients and the enzymes. It's a solvent for transport, where 90% of the blood plasma is made up of water. Regulates the body temperature by sweating, cools down the body using sweating. All of these are importances of water in the human body. State the name of structures L and M. This is the cholera caused by a pathogen in contaminated water. This is the cholera pathogen. What is L? The inner layer. It is the cell membrane and then the cell wall and then the capsule. So L is the cell membrane. But what about M? The black dots are the ribosomes. Cell membrane and ribosomes. Okay. What are two features shown that are typical of prokaryotes? I want some things that are in bacteria. What can I say? Circular DNA. Circular DNA is the loop of DNA. Bacteria does not have a nucleus. They have circular DNA. Okay, they have cell walls. ماشي. I can say they have plasmids, they have flagella. He did not say all prokaryotes. He said this is a feature of the bacteria. Only bacteria have plasmids. What is the type of pathogen type? Cholera is a pathogen. What is the type of pathogen? It is the bacteria. So what is the scientific name? He gave me the binomial name, Vibrio cholera. This is the scientific name. So what's the genus? The genus is the first word which has a capital letter, Vibrio. This is the genus. Okay. Explain how cholera pathogen causes dehydration. This is a repeated question. We will describe the process of diarrhea. How does it happen? Cholera attaches to the lumen, the wall of the intestine. It produces a toxin. The toxin causes secretion of chloride ions. If there is a lot of chloride ions in the lumen, it will have low water potential. So the water moves by osmosis into the gut, into the small intestine, down the potential gradient, causing diarrhea. Complete the sentences about the use of plasmids in genetic modification. During genetic modification, human DNA and plasmid DNA are cut with a restriction enzyme. When we cut them, they create sticky ends. The sticky ends are joined together using a ligase enzyme, forming a new plasmid. The modified plasmid containing the human gene is called a recombinant plasmid. The new one is called the recombinant plasmid. Question 4. Figure 4.1 shows part of the human placenta and the umbilical cord. Placenta umbilical cord. It shows the direction of the blood flow. Okay. The first question he was asking me about what are the functions of the placenta and the umbilical cord? Type. Let's differentiate. The placenta is mainly for exchange. Placenta is where the exchange happens between the mother's blood and the baby's blood. Exchange of nutrients, it takes in glucose, exchange of waste products, the carbon dioxide goes to the mother's blood. Antibodies, they come from the mother's blood. Also, it's a barrier to the pathogens. This is the function of the placenta. طب what does the umbilical cord do بقى? It transfers this stuff. After the exchange happens, the umbilical cord transfers it to and from the fetus. So, placenta is the site of the exchange. It is the barrier to the pathogens, separates mother's blood from the fetal blood. Antibodies are transferred from mother to fetus. طب what does the umbilical cord do? It connects the fetus to the mother, transfers the glucose and the oxygen from the mother to the fetus, transfers the carbon dioxide from the fetus to the mother. It does the transfer to and from the fetus. Okay? Okay. The fetus develops inside the sac. Describe the functions of the amniotic sac or amniotic fluid. The sac produces the fluid inside it and prevents the entry of pathogens from the vagina. Also, the fluid around the baby, it acts as a shock absorber, allows the movement, بيعمل lubrication, so the baby can move inside the uterus, maintains the temperature around the fetus, maintains suitable temperature. All of this will be correct. Syphilis is a sexually transmitted infection that can be passed from mother to her fetus. I did not take syphilis, but I took the AIDS. AIDS is a sexually transmitted infection. What is the name of another sexually transmitted infection? Oh, it is the HIV. The HIV, a virus that is transmitted. What are two ways to control the spread of the STIs? Two ways to control. Anything that applies for the AIDS will apply for the syphilis. Using condoms, testing the blood before transfusion. Okay? Question number 5. Cross-section of a leaf. Cross-section of a leaf. Draw a circle around one vascular bundle. Where is the vascular bundles here? The vascular bundles are in the spongy layer. So I have to find one in the spongy layer. I can circle this one. These large circles are the xylem, or this one, or this one. Yani this is the clearest one. Xylem and phloem. The smaller ones are the phloem. Label line X to identify the palisade. Where is the palisade? Rectangular cells just beneath the epidermis. This is the palisade. What is the name of the cell structure in the palisade where photosynthesis occurs? Chloroplasts. But what is the function of the tissues in the vascular bundle? First of all, I have to tell him what are the tissues in the vascular bundle. Xylem and phloem. Xylem and phloem. And then tell him about what does the xylem do? Transports water and minerals from the root up to the leaves and it supports the plant. What about the phloem? Responsible for translocation, which is the transport of the sucrose and amino acid from source to sink. Function of the phloem, function of the xylem. The leaf shown is from an aquatic plant adapted to live in water. They float on the surface of the water. State the term used to describe the plants. And I said this before. The plants living in the desert is xerophytes. The plants living in water is hydrophytes. Hydrophytes. I have to know its name. Identify the feature T, how it adapts the leaf to float on the surface. Structure T, اللي هو مين? What's structure T? These are the air spaces. How does it allow بقى to float? I have to explain. قول له مثلا it decreases the density. It helps it to float, decreases the density. They call it give buoyancy. This was in the marking schemes before, but if probably you do not know this word, so we can just say that it decreases the density of the leaves because there is air, so it can float on top of water. One other adaptation of the aquatic plants, I can say that the stomata are on the upper surface instead of the lower surface in order to exchange gases with air. One other adaptation, stomata on the upper surface to allow gas exchange with the atmospheric air. The last question, microplastics are pieces of plastic with a diameter less than 0.5. Figure 6.1 shows the mass of microplastics in the ocean between 2000 and 2040. The data between 2000 and 2020 is an estimate. After 2020, it's a prediction. طب why is it an estimate? Suggest. This is a suggest question. You have to think. Why is it an estimate? Any logical reason will be correct. I can say that the microplastics are very small, so they cannot be seen properly. Not all microplastics are collected. They only collected a sample. They are too small to be seen. Any point. You have to think of a valid point. Calculate the percentage increase between 2000 and 2040. You will get the number in 2040. Okay? 1.6, 1.7. And the number in 2000, 0.2, 0.1, 0.2. Okay? And then I will calculate the percentage increase. Final minus original over original times 100. It is 750%. Is it normal to be above the 100? Yes, it's fine. It increased so much. It increased 750%. Phytoplankton are producers. Phytoplankton absorbs microplastics into their cells. What is meant by the term producer? An organism that gets its nutrients by photosynthesis using energy from sunlight. It makes its own nutrients using energy from sunlight by photosynthesis. This is a producer. Take care that he said that phytoplankton absorbs the microplastics. Okay. Figure 6.2 shows a shearwater bird. It feeds on fish in the ocean. Suggest how microplastics can end up in consumers such as shearwater birds. But this is a suggest question. But he gave you a hint. The phytoplankton, which are producers, absorb the microplastics. So when the primary consumers eat the phytoplankton, they will have the microplastics in them. When the secondary consumer eat the primary consumer, they will have the microplastics in them. Why is this? Because they're non biodegradable, they're not digested, they're not absorbed, they just remain as they are in the body cells. This would be the answer. Primary consumer eats the producers, which are the phytoplankton, which contain microplastics. Secondary consumer eats the primary consumer, so the microplastics will move up the food chain as they remain in the body cells. They do not break down. طيب, the rest of the questions. Population of shearwater birds is decreasing. Describe 3 ways that the birds can be conserved. How do I conserve them? Monitor their numbers, education and awareness of the population, captive breeding, protect their habitats, remove the predators. Sub Explain the risks if the population is small. Less genetic variation, less adaptation to the changing environment, increases the chance of recessive alleles being expressed, increases the risk of genetic diseases. This is what they call inbreeding. Inbreeding means that there's a small population and they are all breeding from within each other.